Espera, ¿eh? Ya.
Bueno, tú y yo. Hello, we are the group of Carlos Cañete and Ricardo Ballester, and today we are going to present you the prionic diseases.
So what are we going to talk about today? We have what is a prionic disease. Then I'm going to talk about the epidemiology. Then Ricardo is going to explain you what the Creutzfeldt-Jakob disease is. I'm going to talk about the symptoms. We'll talk about the diagnosis, the types of CGAD. Later we're going to talk about the fatal female insomnia, the German disease. Ricardo is going to explain you what Kuru is. And finally, we'll have the conclusions.
Okay, so first, what is a prionic disease? The prionic disease, also known as a spinal encephalopathies, are a group of fatal and rapidly progressing neurodegenerative diseases that are transmissible between human and other mammals. This group of diseases, caused by prions, um, are caused by prions. These neural proteins has an uncertain function. But we know that in the prionic disease, they acquire the ability to change their own structure into a misfold protein. And also this misfolded structure acquire infectious properties that collapse nervy protein molecules into the same shape. As a result, this abnormal structure form aggregates of proteins called amyloids. This accumulation induces into cell apoptosis and cell tissue damage.
Well, epidemiology. As dangerous as they may seem, prionic diseases have a really low incidence. As we see here, this incidence is about only 1.5 cases per 1 million inhabitants in only a year. So we'd say it has a low incidence. The most common form of prion disease is the Creutzfeldt-Jakob disease, from which 85% of the cases are sporadic. Later we're going to talk about what sporadic means. 10% are familiar forms and 1% are iatrogenic. Iatrogenic, as you may not know this, means that it's a failure from the sanitary system. So nowadays, the iatrogenic cases are quite strange to find, as everything is way more controlled. In the past, patients acquired the disease by having a cornea transplanted or being administered the growth hormone from an infected human.
Okay, so the first disease that we're going to see is the Creutzfeldt-Jakob disease. As we said, the CGD disease is the most common prionic disease. It is a fatal degenerative brain disorder where the prion proteins change their conformation into the misfold structure. It's called the scrappy structure. It usually appears in the age of the 50s, between 50s and 70s.
What are the symptoms of this disease? So we have a rapidly progressive cognitive decline. What does that mean? Well, it will give you anxiety, you'll have change of humors, hallucination, aphasia. Then we'll have cognitive, psychiatric, and conductual disorders, and paired with extrapyramidal and pyramidal syndromes. What are extrapyramidal symptoms? Well, you can see them by making the Babinski. So if it's positive for Babinski, you have these symptoms, right? Pyramidal symptoms, well, for example, losing of appetite, you won't have, you don't want to eat when you have this disease. Sensitive deficits, cerebral ataxia, visual disorder, and with here we can see it's a really deadly disease, because if you have it, you won't last a year.
Okay, so the diagnosis of this disease, in the first place, it's possible to appreciate in the electroencephalogram a general a slowdown of the brain activity. However, in later stages, you can find a characteristic pattern, which is 1 Hz frequency. Then we can analyze the cerebrospinal fluid. in which we can find moderate elevation of protein of the protein 1433 of the 1433 protein. These findings could be supportive in the diagnosis of the CGD disease. However, a positive result should not be regarded as sufficient for the diagnosis. Another protein we can find in the cerebrospinal fluid is the specific neuronal enolase. However, this protein can be elevated in other kinds of dementia as well as in strokes, and it's responsible of the 15 between 15 and 25% of the false positive. Another technique is the magnetic resonance image of the brain. It often shows a high signal intensity in the caudate nucleus and putamen bilaterally on the T2 weight images, especially in the flare and the diffusion sequences. Another criteria of the
Richard? Hello?
the anatomopathological studies.
Can you go back? We didn't listen to you.
Really?
Yeah.
Okay. Which part?
Back to the cranial MRI.
Okay. So the magnetic resonance image of the brain is also a good technique to diagnose this disease. It shows a high intensity signal in the caudate nucleus and in the putamen, bilaterally on the T2 weight images, especially in the flare diffusion, in the flare and in diffusion sequences. Okay, so then another criteria is that it's to don't have other diagnoses. That means that it's the last possible diagnosis. And the definitive diagnosis is to find alterations in anatomopathological studies and in genetic studies. In the anatomopathological studies, we can find the presence of round back walls in the gray matter that gives the spongiform classical appearance. Okay, so it's also very important to remember that it's necessary to alert the CGD cases to the authorities, as the corpse will require a different process.
Well, we're going to talk about the types of CGD. So, starting with the sporadic, which we said it was the 85% percentage of the cases. What does that mean? It means that a random mutation in one of the PPRC proteins can start the illness. So that's it, you don't need anything. It's just a random mutation and there's nothing you can do to prevent it. The second most common one is the hereditary. I can explain you that there are specific mutations on the PRNP gene located on chromosome 20, which doesn't mean much, but it's what it is. Also, we have the acquired one. Acquire means that there's a specific polymorphism on the 129 codon of the 20th chromosome, which predisposes you to develop the disease. Let's continue by talking about the mad cow disease. You probably will have heard about the mad cow disease, but it appeared due to the consumption of bovine meat, which was contaminated by having them. Well, the meat was contaminated because the cows were fed with contaminated sheep. The original sheep had these scrappy proteins. These scrappy proteins wouldn't affect the cows, but the cows were the vector to get to humans. So the cows got infected by the sheep, they didn't notice, and then we ate those cows or something that came from those cows and we got infected. There were over 190 declared cases and it affected people in between 20 and 40 years old. It was curious because it had a slower progression than the normal CGD. it killed you in between 14 and 18 months, which is not much, but it's something. And this variety of CGD was pretty dangerous because it could be transmitted by blood. You can imagine blood donors and people with sharing needles. Finally, I'm going to talk about the iatrogenic. I've already talked about this. It affects patients that have been in contact with nervous tissue, LCR, or blood from infected people, human growth hormone receptors, transplanted corneas, or, as Ricard is going to explain you later with the Kuru, oral consumption of tissues or organs from humans or animals which are affected by the prion disease.
Okay, so now let's talk about the fatal family insomnia. As you can see, it has horrible symptoms. In first stages, you have an increasing insomnia with panic attacks, paranoia, and phobia. You can also have hallucinations, confusional state, dysautonomic disturbances, and severe dementia. Finally, in terminal stages, you get in coma and you die in approximately 15 months. As you can see in the magnetic resonance in the right of the diapo, you can find progressive bilateral thalamic affection, which is a characteristic alteration in the magnetic resonance.
Such a fatal disease, right?
Yeah, horrible.
Well, we're getting to the German Schausler-Schenker disease, which is not really common, but we thought we really should mention it. It has cerebral ataxia and dysarthria, cognitive decline, and starting from 40 years old, and it gets you only 5 years of average lifespan.
Okay, so now the Kuru disease. It's an spongiform disease similar to the CGD, and it's characterized by, well, you can find different stages. First, as well as in many other prionic disease, you have like an asymptomatic incubation period of between 5 and 20 years. Then you start with the disease. The first stage, it's called the ambulant stage because you are able to walk. However, you have ataxia, dysarthria, and decreased muscle control. You look quite like a zombie. Then you enter in the secondary stage, which is the sedentary stage. In this stage, they are incapable of walking. They show depression and emotional instability. You can see in these people an uncontrolled and sporadic laughter, which is very scary, in my opinion. In the third stage, which is the terminal stage, they develop severe dementia, dysphagia, urinary incontinence, and they die in only a few months. This disease was common among the 4 people of Papua New Guinea for eating infected people. It was more common among women and children because they used to eat the brains of the victims, whereas men ate the muscle.
Well, we're getting to the conclusions, and as dangerous and mysterious as this disease might seem, the scientific community is working really hard to eradicate it. But till then, we need to resign and hope that we are not that 1 in a million person who contracts this illness. Remember, folks, don't eat anyone's brain. Thank you very much for listening.
Goodbye.
Bye.